Hello guys, welcome to the knowledge domain, the concept of life sciences and biotechnology. In the previous lecture, we have learned the electron transport system where the electron is donated by the NADH and the final receiver is the oxygen atom and result in the water molecule. So we also learn comparatively the way the light energy involve transfer of the electrons from the water molecules and finally the NADPH is formed. In this lecture, we are going to learn the photosynthesis process in slightly more detailed way. So before going into the topic, we should know that in the present day, our Earth atmosphere is having almost 21% of the oxygen, but this was not the case 3 billion years ago. So the arrival of the cyanobacteria results in the photosynthesis by which there is the release of the oxygen, and at the same time there is the antioxidant defense mechanism evolved. So a lot of organisms who do aerobic respiration, they also collaborate with the increase in the oxygen amount in the atmosphere. So we all know that whatever energy the entire living kingdom of this earth is using, the source is the solar energy or the light energy. So it is captured, the trapped by the photosynthetic cells, and whatever energy is trapped there, that is being used by the plant system as well as the remaining all the types of the creatures, heterotrophs as well as the autotrophs. So this is a very simple equation where the carbon is present in the atmosphere as the carbon dioxide, and the source of the electron is the water. And in the presence of the light, the very high reduction potential water molecule is able to donate its electron. So the result is the oxygen molecule, and this is the final result is the carbohydrates. So in the light reaction, the light, in the presence of the light, there is a means energy is trapped in the form of the reducing substance like the NADPH and the ATP, and this chemical energy is used in fixing carbon dioxide to prepare the carbohydrate. That is called the carbon assimilation reactions. So this also, this is a chloroplast. This is the plant cell. In this plant cell, there are chloroplast, and this is a double layer. And for contrary to the mitochondria, the electron transport system is not present in the inner membrane. It is present in a further means an subcellular sub-organelle. Inside the organelle, there is a suborganelle structure that is called the thylakoid, and there is a lamellae in the thylakoid. So in this thylakoid membranes, the electron transport system is present, the chlorophyll molecule is present that actually absorbs the sunlight and it traps the energy in the form of the NADPH and the ATP. So in the Robert Hill, 1937, he extracted the green pigments or the chlorophylls, and he found that there is an increase in the oxygen in that plant extract or the leaf extract, and he formulated this equation that the water, and this is the electron acceptor, this is the electron donor, this is the electron acceptor, and finally the oxygen is released in the presence of the light. And further, subsequently, the Severo Ochoa, he actually delineated this formula and find out that what is the electron acceptor, that is the NADP plus. In the presence of the light, it is reduced into the NADPH, and these protons are released, which actually moves from the stroma into the, it moves from the stroma of the chloroplast into the thylakoid lumen, and the oxygen is released. So this also we have discussed in the last lecture. So here the important point is this is called the dark reaction, but the most appropriate terminology is the carbon assimilation reaction or the carbon fixation reaction, because the atmospheric carbon dioxide is fixed or it is used in preparing the glucose or the carbohydrate. And in this, one Einstein or 6 into 10 to the power 23 photons of visible light equal to the 170 to 300 kilojoule according to the Planck's equation, E equal to h nu, where h is the Planck's constant that stands for 6.626 into 10 to the power minus 34 joule second. So this is the chlorophyll molecule. It is a polycyclic planar structure resembling the protoporphyrin of the hemoglobin. There is only one difference is in the hemoglobin here the iron molecule is present Fe2 plus, and here it is magnesium. So this is the chlorophyll A formula, and chlorophyll B, instead of CH3 there is a aldehyde group, that is the chlorophyll. And this bond is saturated in case of the bacteriochlorophyll. It is a saturated bond. And in case of the bacteriochlorophyll, along with this saturation bond, this CH2 group is actually ketonic group. And there is a phytyl side chain in this case. And this is the series of the light. This is the chlorophyll A reaction center. So we have 1, 2, 3, 4, 5, 6, 7 photo absorbing molecules are here. The PE stands for phycoerythrin. This is phycocyanin, and this is yellow phycocyanin. And the light 480 to 570 is absorbed here, and then it is transferred into the chlorophyll molecule. And on this series, this is light from 550 to 650 nanometer absorbed and transferred to here. And the light harvesting complex, this is the light harvesting complex. It has the accessory pigment like the beta-carotene, which is a yellow carotenoid lutein that absorbs light at the wavelength not absorbed by the chlorophyll molecule. So in this way, it is actually complementing in augmenting or optimizing the light absorption ability of the light harvesting complexes. So this we have seen in the previous lecture. Here we have the photosystem 2 and the photosystem 1. So here this is the reaction center that absorbs light at means 680. In the photosystem 1 it absorbs light at the 700 nanometer. And this photosystem 2 is like the one pigment of the purple vector, that is the pheophytin quinone type of system. And in this photosystem 2, the chlorophyll A and B are found in the equal proportion. But in the photosystem 1 is like the photosystem of the green sulfur bacteria. In this one, the chlorophyll A is found more in quantity than the chlorophyll B. Okay. So we know the reduction potential of the water. This is 0.816 volt, which is very high reduction potential. So it does not have the ability to release its electron or donate its electron. So this ability is taken by the getting of the light. Okay. So when the light, it gets the energy from the light, the electron get excited, and it's through a series of uh, through a series of the exciton, means the energy present in the one molecule that transfer into another molecule having the slightly low reduction potential, slightly higher reduction potential, so that it has the ability to accept the electron. So this is one exciton. So it is through a series of excitation reduction process, the electron travels. And during this process, the energy utilized in pumping the proton and generating the ATP. So this is the pH used the pheophytin. So it is having the low reduction potential compared to the PQA, subsequently PQB. So if the reduction potential increases, this is the reduction potential in the volt. So this is the lowest, this is the highest. So it is the electron is moving on a step-by-step process. So in this process, the energy is released and that is being used in pumping the proton. So PQ is the plastoquinone A, plastoquinone B. This is the cytochrome B6F complex. Okay. And plastocyanin is a water-soluble protein molecule that is equivalent to the ubiquinone of the mitochondria. And from here, the electron moves into the reaction center 700. And when it gets the light again, the energy, again it gets the electron is excited and the reduction potential it means get reduced so that it can again move step by step process, and in this process the energy is released. So this is the electron acceptor, the chlorophyll 0, this is the phylloquinone, this is the iron sulfur protein, this is the ferredoxin, and this is the ferredoxin NADPH oxidoreductase. Finally the NADPH is reduced. So whatever energy is released in this process, it is used in two forms. One is forming the proton gradient, another is trapping the light energy into a chemical energy in the form of the NADPH. This is a reduced substance. And this plastocyanin is a one electron carrier. So in one process, there are two electrons released. So we need the two plastocyanin. So for O2, it is a, if you see this on H2O, this is one atom. So we need the two water molecule to make an O2. So transfer the four electrons from the two water molecules. So from the one atom, there are the two electrons. So from two water molecules, there are the four electrons. So if it is the O2, means from the two water molecule, we can generate two NADPH finally. And so in the transfer of the one electron, there is the two photons needed. So for the four electrons, there is a need of the 8 photons. So one photon here in the photosystem 2 or the reaction center P680, another photon at the reaction center P700. So 8 photons, one O2 molecule is generated, and there is a release, there is the use of the two water molecules. So that is the stoichiometry. So two water molecules, two NADP plus 8 photons equal to one oxygen molecule plus two NADPH plus two H plus. So this is the light reaction, and this is a comparison. We have done a comparison before also. So this is another comparison. So this is the photophosphorylation. So in this process, there is a phosphorylation of the ADP into the ATP, which is the energy currency. That is possible because the movement of the proton along the concentration gradient through the ATPH. So this is a photosynthesis of the photophosphorylation. And in the mitochondria, this is the oxidation process. Because of that energy is released, so it is oxidative phosphorylation. So this is NADH dehydrogenase in case of the mitochondria. In this case, we have the photosystem 2. So in this case also, this is quinone, or means it is reduced to quinone. In this case also, it is plastoquinone. So in this case, it is the complex 3 cytochrome C oxidoreductase. In this case, it is the cytochrome B6F complex. So they have a similar function, it is almost similar. This is the complex 4, that is the cytochrome C oxidoreductase. And this is in this case, it is the photosystem 1. So this is the finally the F0F1 complex or the ATPH. So in the oxidative phosphorylation from the intermembrane space, it is moving into the stroma. It is moving into the matrix of the mitochondria, and in case of chloroplast, it is moving into the stroma of the chloroplast. So this is the sequence of the oxidoreductase process in which the electron is moving from the low reduction potential to the high reduction potential having molecules. During this process, the energy released is used in pumping the proton across the concentration gradient to create the proton motive force. that finally move through the ATPase to generate the ATP. So in case of the photosynthesis, the energy that is released in the form of the NADPH or the ATP that is used in fixing the CO2. So how it is happening? This is the ribulose 1,5 biphosphate. So this is the molecule. So in the dark reaction or the carbon assimilation, dark is misnomer, the one CO2 molecule, it is taken by ribulose 1,5 phosphate, which is a 5 carbon molecule. So in this form, we have the 6 carbon molecule is possible, but the uh, product is not so stable. So immediately it split into two, 3-phosphoglycerate. So they can be used in a Kelvin cycle, or one of the 3-phosphoglycerate can be utilized in the gluconeogenesis or the other pathways. So in one Kelvin cycle, we can fix, or the plant can fix one carbon molecule. So to prepare a glucose, which is a 6 carbon molecule, there is the need of the 6 Kelvin cycles. So these 3-phosphoglycerate, it utilizes an ATP to generate 1,3-biphosphoglycerate. Okay, but there are two 3-phosphoglycerate. So here there is a utilization of the 2 ATP, and there is already one utilization of the ATP here that convert the ribulose 5-phosphate into the ribulose 1,5 biphosphate. So there is a utilization of the 3 ATP, and this 1,3 biphosphoglycerol it become the glycerol 3 phosphate by the use of utilization of the 1 GAPDH, the NADPH. So for the 1,3-phosphoglycerate, 1 NADPH. So for the 2, there is a 2 NADPH. So this is one Kelvin cycle, 3 ATP, 2 NADPH. So for one glucose molecule that is multiplied into 6 Kelvin cycles, so there is a need of the 18 ATP and the 12 NADPH to prepare one glucose molecule. Okay. So this is to understand in detail that we know ribulose 5-phosphate become biphosphate use of the 1 ATP per Kelvin cycle. So in the 3 Kelvin cycle, there is a need of the 3 ATP. Okay. So this ribulose biphosphate, one ribulose biphosphate utilizes one CO2. So 3 ribulose biphosphate will use the 3 CO2. So here there is a 6 ATP, and this utilizes 6 NADPH, but this is half of the glucose. This is becoming 3 because considering this product, which is 3 carbon molecule product, the GAP is a 3 carbon molecule product. So to make a glucose unit 6, so everything will be multiplied into the 2. So finally this is a 3 plus 3 equal to 6. Then this is 3 plus 6 equal to 9. So this is 5 plus 4. Then again this is 4 plus this is 3 equal to 7. This is a 7. This is a biphosphate. If one phosphate is released, it becomes a monophosphate. So this is still 7. 7 plus 3 equal to 10. So this is 5 and 5 equal to 10. And this 5 becomes 6 by fixing one CO2 molecule here. Next is the photorespiration. Because this RuBisCO, which is a very important enzyme, the carbon fixation. So this is not so efficient, and this is the enzyme that is found in the highest abundance in this living world. So in that captures CO2 from the air that we have discussed in the earlier slide. Not very good at distinguishing oxygen from the CO2. So 1 out of the 4 times they usually take the oxygen instead of CO2. So instead of carbon dioxide, oxygen is taken. So if the oxygen is taken, 5 will still remain 5 carbon. RuBP, ribulose biphosphate, right? So this is 5. So instead of getting the 6 carbon intermediate, this 5 will split into this 6 should have been split into the 3 PGs. Okay, so if it is 5, so there will be 1 PG means, and Next one is the 2-phosphoglycolate. Okay, 3 plus 2 equal to 5. So photorespiration that convert this 2-phosphoglycolate into the 3-PG because this is toxic. And in converting these 2 carbon molecule into the 3 carbon molecule, there is the utilization of the 12 ATP per molecule of 2-phosphoglycolate. So this actually cost plant lot of energy, but plant captures CO2 from the air, combining it with the RuBisCO to produce the 3-PG. So creates a 3 carbon molecule. These plants are called as C3 plants because they are converting these 2-phosphoglycolate into the 3-PG. So these are the C3 plants because they are using the 3 carbon molecules. Okay. So the next one is this is the C4 plants. Why they are C4 plants? Because considering the lacuna of the RuBisCO, so in this plant, what happened? There is a additional cell that is called the mesophyll cell. So this cell actually it traps the CO2 by the enzyme that is the phosphophenol pyruvate carboxylase. So this enzyme actually they fix the carbon so that it will reduce the boron and the RuBisCO. So they convert this and they make a oxaloacetate, which is a 4 carbon molecule that convert into the malate, and this malate goes into the bundle of sheath cell that is adjacent to the mesophyll cell. And in this sheath cell, this malate it releases the CO2 so that there are the enough CO2 and there is the less exposure to the oxygen. So this RuBisCO enzyme now it is more efficiently bind to the CO2, and like this it avoid in preparing the 2-phosphoglycolate by misjudging the O2 and binding to the O2. And there is the another type of plant that is called the CAM, Crassulacean acid metabolism plant. It is found in very hot and dry places. And the plants are like orchids, cacti, and the pineapple. So in these plants, what happens? This process is almost like the same here, except this mesophyll cell. In this plant, there is a stomata present, and these stomata are closed during the daytime, and in the night time they take the CO2, and in the daytime they close it so that during the daytime when the light is available, there is no intake of the air because the stomata is closed, so that the RuBisCO enzyme it is surrounded by more CO2 molecules instead of the oxygen molecule. So like this in these plants, they increases the efficiency and the specificity of the RuBisCO by properly utilizing the stomata molecules. The stomata, which actually closes the pores present in the leaves. So whatever till now we know that we get from the plants is the oxygen and the carbohydrates, but there are some other metabolites that also we get from the plants. These are called the secondary metabolites like the alkaloid. One of the examples, there are 3 families of the secondary metabolites. So one is the alkaloid. One example is caffeine caffeine is an alkaloid, which is a methyl xanthine chemical name, and that is a derivative of the purine compound, and it derived from the purine nucleotide, and it works an antagonist of adenosine because what happens in our brain, there is a receptor. In that receptor, if adenosine will go and bind, then we feel sleepy. So what happens if caffeine almost looks like the adenosine, so it will go and bind to this receptor so that the adenosine cannot bind, so that we don't feel asleep. And the next family is the phenolic compounds like the vanilla and the salicylic acid, and it is like the derivative of the aspirin. And the third family is the terpenes and the terpenoids. They are from the resins from the evergreen trees, and it is also derived from the mevalonate, like cholesterol is derived. And one of the examples the carotenoid. Thank you.